Hello dear students and friends. How are you all? Welcome to Karch Agri channel. If you are not a subscriber, please subscribe right now. Thank you. Today we will discuss about classification of Chytridiomycota Zygomycota and important characters, symptoms, life cycle of Synchytrium, Rhizopus Mucor. Chytridiomycota first discovered by Bartnicki-Garcia, 1970. This comes under true fungi cell wall as chitin. It is a transitional group between Protista and fungi, production of motile zoospores. Basal position in fungal tree. Sexual spore-resting sporangium. Sexual reproduction through Planogametic copulation. Symptom of Synchytrium endobioticum, cause the wart disease in potato. Symptoms of the disease appear only on underground parts except roots of the plant. Characterized by warty, tubersome dirty cauliflower-like outgrowths on infected parts. The warty outgrowths vary in size from small protuberances to large intricately branched systems. Early in the growing season they are green or greenish-white in color. If exposed to light but are cream-colored or black on underground parts. On the tubers, mostly the warts are larger than the tuber itself, covering the whole tuber. In advanced stages, the warts become dark black in color and may sometimes be attacked by saprophytic fungi. The wart usually consists of distorted proliferated branched structures grown together into a mass of hypertrophied tissue. In case of Phylum Zygomycota, Thallus well-developed, branched mycelium. Coenocytic hyphae. Cellular organelles present, centriole absent. Cell wall cytosin chitin. Rhizoid anchoring organ. Intervening hyphae connecting two group of rhizoid called as stolon. The symptom of Rhizopus is Rhizopus rot is a common disease of jackfruit flowers and young fruit. It causes a soft, watery, brown spot on the fruit, and soon becomes covered in a gray-brown, later black, furry mold. Fruit symptoms occur on the tree and in storage. Now we are discussing about mucor. Mucor also refers to black or bread mold. It belongs to the class of Zygomycetes. For most of the mucor, the mode of nutrition is saprophytic, grows in the dead decaying matter, and for others, it is coprophilous, grows in cow dung or the dung of other herbivorous animals. Mucor grows on a variety of substrates like bread, jam, jellies, vegetables, etc. The absorption of nutrition is through the mycelial surface or hyphae. The vegetative body of mucor is eucarpic, because in this the only thallus differentiates into the reproductive structure. The major reserve food material is in the form of glycogen and oil droplets. The cell of mucor composed of mainly cellulose and chitin. The colony of mucor shows rapid growth. The color of the colony is usually white to gray and turns to brown when the culture becomes old. hyphae, coenocytic and branched. Spores, generally black in color, but can vary with different species. The spores can be motile or non-motile and can exist in variable shapes. Thank you.
Asexual reproduction. These are the spores formed within the cell or sporangium and are non-motile. From the hyphae, first sporangiophores arise singly and are erect in position and unbranched. Then, maturation of sporangiophore occurs where the cytoplasm and nuclei push upwards by making the aerial hyphae swollen from the apical end. After that, it develops a large round sporangium. During this maturation phase, sporangium differentiates into spiroplasm. It is thick, dense, multinucleate and present inside the sporangial wall. Columellaplasm, it is vacuolated and nucleated towards the centry. After this, a number of small vacuoles appear between these differentiated portions. The space between the vacuoles forms cleavage furrow. Then, to the inner side of cavity septum forms that further divides the inner columella and upper spiroplasm. This septum then grows to form a dome shape and push itself into the sporangium. Cleavage occurs in the spiroplasm between the nucleus and the cytoplasm. This division forms a wall around many thin-walled, multinucleate spores called sporangiospores. The sporangiospores then releases out of the sporangia, when columella swells up, which creates pressure on the sporangial wall, cause cell lysis. The spores remain dormant for some time and, when they obtain suitable substratum, they germinate to a new vegetative body through germ tube. These spores are covered by a hard wall that forms inside the vegetative cell during unfavorable conditions. Let's see something about chlamydospore. In unfavorable conditions, mycelium becomes septate by the accumulation of nuclein cytoplasm in a certain portion surrounded by a thick wall forms chlamydospores. This spore then detaches from the mycelium and remains dormant. On favorable conditions, they form a germ tube. Sexual reproduction. First, the thallus of two opposite strains, that is, one is plus and other is dash, comes in contact with each other. When they come in contact, there develops a small outgrowth or protuberance from both of the thalli. After that, the outgrowth swells to form progametangium. Then septum develops between the progametangium and the fusion of progametangia occurs, which results in the formation of gametes, refers to Coenogametes. Then gametes of both the strains fuse with each other to form zygote. The zygote then enlarges in size and gets surrounded by a thick-walled structure called zygospore. Zygospore is dark black in color, which develops and get covered by two layers, namely, outer layer, also refers to exosporium, inner layer, also refers to endosporium. The zygospore remains dormant for some time, and on favorable conditions, promycelium develops out from the zygospore, forming a new vegetative body. Hey everyone! Thank you for watching. If you are not a subscriber, subscribe now. Thank you. See you later. Bye.